so our next speaker is from Stanford. I'm going to introduce Tushar Desai, who's going to talk to us about the epithelium as guardians in the lungs. Great, thank you very much.
Is this the advanced?
Yeah, this one here. I'll just take this one.
Is that a pointer? Or?
You have a pointer here.
Pointer here. Great. Okay. Thanks. Good afternoon. Don't let Victor get you down. Let's switch topics to something more regenerative. Sorry. Okay. So anyway, I want to thank the organizers, Tim and Lisa, for inviting me to speak, and I'm going to tell you a nice story about epithelial repair in the lung. So I want to just start by talking about, every tissue in the body has distinct stem cell needs and distinct constraints. So I just want to talk about first the blood system. So this is one of the most well-characterized stem cell compartments in the body. And so here you've got stem cells that reside in the bone marrow in their niches, and they can give rise to daughter cells that can then go on and proliferate extensively and generate all of the different lineages of the blood system. So here you've got a regularly turning over tissue, and since these cells are mobile, you don't have to deal with the constraint of trying to organize the differentiated cells properly within the structure. So that's sort of maybe an easier task than other tissues. If you look at the intestine, so the intestine is a continuously cycling tissue, but it's a solid tissue, so you have to have a way to integrate all the differentiated cells properly. And so you have these stem cells that reside at the base of the intestinal crypts, and they are active, and they continuously cycle and differentiate into transit amplifying cells, which then generate the mature intestinal lineage cells like Paneth cells. And this mechanism here is the replacement, because it's an intact epithelial sheet that wants to remain intact, is that you basically sort of have an escalator. You've got the old cells at the top that slough off into the intestine, and they're just continuously renewed by these cells that move up from the crypt. Now the lung is another different tissue. So the lung, unlike the intestine, the turnover in the lung of the epithelium is extremely slow. And so but yet you, like the intestine, you have an intact epithelial sheet that maintains an important barrier function. And so I'm going to first tell you about, this is some work that we did some years ago where we looked at the contribution of type 2 cells to generate new type 1 cells during aging. And what we found is that here, when we genetically marked all the type 2 cells in green and the non-type 2 cells are in red, is that initially type 2 cells do not give rise to type 1 cells in development. You can see that you have green type 2 cells, but no green type 1 cells. Then as we looked at progressively older ages, we started to see the appearance of these foci, where you can see here there's a green type 1 cell attached to a type 2 cell, and here's about 3 type 1 cells there in the focus. And over time, these foci increased in size, but they did not really increase in number. I'm not showing you the data in the interest of time, but these foci are also monoclonal. So an interesting feature of these foci is that even though there are many type 2 cells in the area, only one type 2 cell gave rise to this entire clonal focus. The other features that were a little bit surprising, at least to me, were that the turnover is very patchy. So we saw these foci preferentially in subpleural and perivascular regions of the lung, and it was very inhomogeneous, very reminiscent of the distribution of disease in IPF, actually. So just to summarize here, type alveoli are renewed by these rare type 2 cells that make up about 1% of the population, and they're intermittently active, and they generate these expanding foci, what we call renewal foci, during aging. Another interesting feature is that unlike in the intestine and the blood system, type 2 cells are not dedicated stem cells, meaning they have other functions. In the intestine and the blood, stem cells only exist to make new cells. But as far as we can tell, type 2 cells in the lung, the ones that act as stem cells, they also participate in surfactant physiology. So we refer to these as a bifunctional stem cell because they're constitutively executing both stem cell and a physiological function. Okay, so this is just summarized here. At the time that we had done those studies, this was our working model, is that you have transient, whether it's programmed or stochastic cell loss, and then a type 2 stem cell will duplicate and either remain as a type 2 cell or generate a type 1 cell. And based on other data that I'm not going to show, we believe that the trigger for this proliferation was an EGF ligand that was activating, working through EGF receptor and KRAS. So at this time, we were wondering, is there a way that we can identify this 1% prospectively? In other words, are they different somehow than other type 2 cells? Do they express a special marker? And what regulates their stem cell activity? And furthermore, what determines following the differentiation, the duplication event, whether the progeny would remain as a type 2 cell or transdifferentiate into a type 1 cell? So I'm going to talk now about Wints for the rest of the talk. So Wints are a secreted family of ligands that are hydrophobic, and so they act very, very short range, maybe across a distance of 1 or at most maybe 2 cells. And Wints mark stem cells in a variety of tissues. So Wnt-active stem cells are, for instance, the intestinal stem cells that I mentioned to you. They express LGR5, which is a transcriptional target gene of Wnt. And they also express AXIN2, which is another target gene of canonical Wnt signaling activity. So what we observed when we used a genetic strategy to mark the AXIN2 expressing cells in the mouse lung is that about 1% of them are actively undergoing Wnt signaling. Now, when we looked at the behavior of these cells, we could see that you could sometimes catch them in the middle of mitosis. So this is a type 2 cell that is expressing the green lineage mark of an AXIN2 expressing cell as in the process of dividing. Here's a focus of type 2 cells where you can see 3, I guess this one is dividing, and then you've got another daughter type 2 cell over here. And they also give rise to type 1 cells. And so you can see here's a type 2 cell that's SPC positive, and it's expressing the lineage tag, and then it's given rise to this type 1 cell that is adjacent to it. And when we quantify this over a 1-year period, we see that the percent of AT2 cells that are in the AXIN2 lineage increases from about 1% up to about 6%. So while the magnitude of this increase is not particularly large, it is significant, we think, given the very low rate of cellular turnover in the lung, so that these cells are disproportionately, excessively contributing to new type 2 cells. So we were pretty excited by this, and so we wanted to know what is the source of the Wnt. I told you that AXIN2 expression means the cell is receiving Wnt signal, and Wnt is a very short-range signal, so there has to be a cellular source of Wnt very close by. Now, we looked at various cell types, but we were particularly interested in fibroblasts because it's been classically known that there are very intimate physical associations between type 2 cells and fibroblasts that people had noted on electron microscopy many years ago. So here we we used single-cell RNA sequencing to sequence fibroblasts from the adult mouse lung, and we took a look at all of the Wnt ligands, and we asked, are there any Wnt ligands expressed by fibroblasts, and if so, which ones? And you can see here that notably there's a significant minority express Wnt5A, and then a smaller segment express Wnt2B. So we said, okay, if we're correct, then if we stain for Wnt5A, SPC, and AXIN2, we should see that the AXIN2 positive type 2 cells will be next to a Wnt5A expressing cell, and the AXIN2 negative type 2 cells will not. And so that's exactly what we found. This is an in situ hybridization, and I'm just going to blow up the images here. So this is an SPC positive type 2 cell that is not near a source of Wnt, and it is AXIN2 negative. But when we saw AXIN2 positive type 2 cells, AXIN2 is here in green, they were right next to Wnt5A expressing cells, and I don't have the fibroblast co-stain, but when we in this image, but when we co-stained for fibroblasts, they were PDGF receptor alpha positive. And this is just quantified here. So this was very exciting. So it sort of, we had developed a model here that we have these rare type 2 cells that exhibited the stem cell activity, and that they were juxtaposed next to fibroblasts expressing Wnt5A. And so really this was quite interesting. You have these distributed 2-cell signaling centers throughout the lung. Now, this is one of the remarkable things about the lung. I was mentioning about the different stem cell needs. So from birth until death, the lung is continuously exposed, obviously, to the airborne environment. And so, even though there's very slow turnover with aging, humans can get viral infections causing acute cell death, toxic inhalations that can cause epithelial cell death, and pneumonias and other scenarios where you can acutely lose a lot of alveolar epithelial cells that you need to repair. So, and this is a really nice classical study where in rodents that are susceptible to hyperoxia, Adamson and Bowden did some very carefully timed radiolabeled thymidine incorporation studies where they could mark and visualize proliferating and dividing cells, and they did a time course, and they found that when you injure the mouse lung with hyperoxia, which results in death of many type 1 cells, you would immediately, the type 2 cells would start proliferating, and then after a couple days, they would start dividing, and then they could trace out the thymidine, the proliferation lineage mark in type 1 cells. And so when they did these studies, they saw that there was widespread type 2 cell proliferation. It wasn't these rare cells that I was talking about. So, and this is just represented here that unlike with aging, with an acute injury, you can recruit what I'm just going to refer to as bulk type 2 stem cells, or you could call them facultative type 2 stem cells, that they are only active under conditions of injury. So under aging conditions, you have these rare type 2 stem cells that are intermittently active, and most of the other type 2 cells appear to be fairly quiescent. But then with an acute injury, all of a sudden, they get harnessed to proliferate and help facilitate the repair. So we next wanted to know, okay, so we saw that, that there's this 1% of type 2 cells. It's marked by axin 2, and it maintains its status by receiving Wnt5a from an adjacent fibroblast. And I'm going to show you this data later, but we found that when a cell divides and the daughter cell goes further away from the Wnt source, it will transdifferentiate into a type 1 cell. So in addition to maintaining this, marking the stem cells, the Wnt's also regulate its differentiation status. So we wanted to know, okay, well, with acute injury, when many type 2 cells begin to proliferate, is this also have to do with the Wnt pathway, and also are the fibroblasts just churning out more Wnt's? What's going on? So here we turn to our good friend diphtheria toxin, which is a way that we could really selectively ablate the epithelial cells without perturbing the niche. And so here what you're looking at is, this is a mouse in which the recombined cells that are expressing the diphtheria toxin receptor are in green, and the unrecombined cells that did not express the receptor are in red. And the green cells here, because we used a driver that expresses only in the epithelium, these are all the epithelial cells, and these are all the non-epithelial cells. So we tuned our dose of diphtheria toxin to cause a pretty significant ablation without resulting in death of the animal. And so you can see here, 18 hours after administration of the diphtheria toxin, you can see that many of the epithelial cells have been ablated, whereas the mesenchymal cells are unaffected, as expected. And then if you look 5 days after tamoxifen or diphtheria toxin administration, you can see that the epithelium has repaired. So we then said, okay, in order to test whether is Wnt signaling important for this repair, let's block Wnt signaling and then do the injury. And when we did that, chemically here, using C59, which is a compound that blocks the ability of cells to secrete Wnt, you can see that repair is inhibited. So we were very excited because this suggested that Wnt is important not just for the rare stem cells, but it's important for the repair following an acute epithelial injury. Now, this experiment, we're blocking Wnt in every cell, not just in the type 2 cells. So we wanted to look further and ask, was Wnt active in the type 2 cells, and was that responsible for this effect? So here we did, again, some in situ hybridization and some antibody staining. So here you're looking at the lung before injury. So Ki67 is stained in white, and you can see that, as expected, the cells are quiescent. They're not proliferating. surfactant protein C, the type 2 marker, is in red, and axin 2, the Wnt target gene that indicates activity of the Wnt pathway, is in green. And as expected, the majority of the cells are not proliferating, and the majority is also not expressing axin 2. And then we looked following the ablation, and you can see pictures here, and the quantitation on the right is that following the injury, many type 2 cells begin proliferating. You can see the Ki67 come on, and it's upwards of 80% of the type 2 cells enter the cell cycle. And then you can see here the axin 2 and the SPC. You can see that about 75% of these cells, we can distinguish axin 2 expression. So we're very excited because this shows that with the acute injury, the Wnt signaling is being received by the type 2 cells and is coincident with their proliferation. And then we quantified what happens with the C59 with the inhibition of Wnt signaling. And you can see that there's a significant reduction in the proliferation and the axin 2 expression in the type 2 cells. Now we wanted to know, okay, what is the source of the Wnt? So now you go from having these rare fibroblasts and these rare type 2 cells to all of a sudden almost every type 2 cell that has survived is axin 2 positive. So there has to be a nearby cell expressing Wnt. And so we first asked, is it Wnt5a? Is it just that there's more cells turning on Wnt5a? And it was not Wnt5a. There was no difference in Wnt5a with the injury and without the injury. So at this point, we were a little bit unhappy initially because there's so many Wnts that we didn't know which one to look for as a candidate for the Wnt that was active. But then we took a page from the developmental biology playbook, and it turns out that Wnt5a and Wnt7b are the two Wnts that are developmentally expressed. And because our lab is right next to Roel Nussi's lab, who discovered Wnts and has been working on Wnts for 30 years, they also recommended that we look at Wnt7b because of some other experiments they had done with injury. So we said, let's do that, and so we did that. So this is in the uninjured state. Again, you can see the type 2 cells in red, and they are not expressing Wnt7b. And then with the injury, you see that these are the type 2 cells, and here's the Wnt7b. Now this was quite interesting because it turns out not only are the type 2 cells Wnt active, but they themselves are the source of the Wnt. So the Wnt7b is not coming from any fibroblasts. It's being produced by the type 2 cells themselves. And you can see that over time, as the epithelium repairs, it comes on very, very quickly, and then it just goes down, and you end up back at the basal state where you have the rare type 2 cells that are Wnt active. So it's remarkable that not only the identity of the Wnt ligand changes, but the source of the Wnt ligand changes. So you go from a juxtacrine niche that regulates these type 2 cells to an autocrine niche. So we also purified the cells and did quantitative RT-PCR, and we found that not only Wnt7b, but actually there was a suite of Wnts that begin to be expressed by the injured, by the type 2 cells in the context of injury. And when we looked before injury, no Wnts were expressed. Okay, now with this acute injury, for all we know, there could be other Wnts expressed by a lot of other cells, for instance, immune cells or other cells in the mix, and maybe although the type 2 cells are expressing Wnts, maybe they're also receiving Wnts from everywhere. It's just a complete Wnt chaotic situation. So we wanted to know, is the source, the bioactive source of the Wnt, is that indeed an autocrine signal? And so we took advantage of another genetic strategy here where there's a gene known as Wintless, which when you delete Wintless, you cannot secrete Wnt. You can still receive Wnt, et cetera. You can respond to Wnt. You just can't make it any longer. So we did another injury experiment where we infected, used a lentivirus, an adenovirus that infects type 2 cells, and we used it to delete Wintless in the type 2 cells. And this adenovirus also expresses a green fluorescent protein. So you can see, for instance, here is a type 2 cell in which Wintless has been deleted. And when we stained for type 2 markers, the GFP and the proliferation marker, we found this anti-correlation. So it was quite clear that here, if you look at the percent of cells that are Ki67 positive, it's very significantly reduced, specifically in the GFP expressing population. So this really suggests that not only is the type 2 cell expressing the Wnt, but that is the sole source of the Wnt that regulates its activity. And so this is just schematized here that you have under aging conditions where there's very slow turnover. You have these, this 2-cell niche, and with acute injury, you get these autocrine niches that are distributed throughout the lung that can facilitate this proliferation and repair. So you've got this juxtacrine-rich rare AT2 cells, and it's these subpleural and perivascular foci, but then with the injury, it's this autocrine niche where many type 2 cells proliferate and they're diffused throughout the lung. Now, I'm just going to end with another interesting thing. So, you know, in many tissues, Wnt actually drives proliferation. And so we asked, you know, in our system, is Wnt driving the proliferation? And we found that we tested this both in vivo and in vitro. And here's a culture system where we took type 2 cells from the mouse lung, and we cultured them, and we assessed their proliferative response to different factors. So when you add Wnt5a or Wnt3, you see a slight but not statistically significant increase in their proliferation. Now, when you add the EGF ligands, you can see that there is a statistically significant increase in proliferation. But when you add the combination of the Wnt and the EGF, you see a massive increase in the proliferation. So Wnt is not directly driving proliferation of these cells, but it is enabling them to be responsive to the EGF ligand and promoting their ability to respond to this proliferative cue. The last experiment I'm going to tell you about is, because there are such good genetic tools for the Wnt pathway, because it's such an important pathway, we also did some in vivo experiments. We said, okay, let's take a mouse and we'll label all the type 2 cells like I had showed you earlier, and there's a certain proportion that that's stereotype where the type 2 cells will proliferate and generate a type 1 cell. And so you can see a yellow type 2 cell here and a green type 1 cell. And we said, okay, let's both delete beta-catenin, which will abrogate the ability to undergo active Wnt signaling, and let's do another experiment where we will express a stabilized beta-catenin. So the stabilized beta-catenin experiment will make that cell unable to turn down Wnt signaling. So when we looked in the first case, we saw that there was a dramatic increase in the number of type 1 cells. Now, the type 1 cells we saw also, it was interesting, they were not associated with a parent type 2 cell. So in other words, you can see here there's a green type 1 cell, but no green type 2 cell. And we saw this repeatedly, whereas with turnover, you would see that these were following a proliferation event. So we concluded that when acutely abrogating Wnt signaling in these type 2 cells was just making them directly transdifferentiate into a type 1 cell. Now, when we did the opposite experiment, we prevented the type 2 cells from being able to turn down Wnt signaling. We saw the opposite effect, which is that it blocked their ability to generate type 1 cells. And this is just quantified here. So our conclusion is that Wnt is really, it's really conferring stemness to the type 2 cell population. It's not a proliferative driver, and it's not, but it's required for proliferation. And so we really think of it that it's sort of maintaining this stem cell state is how it's acting. So just to summarize, I told you that repair of acute alveolar injury depends upon these facultative type 2 stem cells that are transiently activated, and they're activated by expanding and changing the Wnt niche. Wnt endows type 2 cells with this alveolar regenerative capacity. It's required for that. Downregulation of Wnt signaling is required for type 2 cells to transdifferentiate into type 1 cells. And we're very interested in looking at the role of dysregulated Wnt signaling in IPF. So we've heard a lot about hyperplastic type 2 cells, and many studies have shown elevated Wnt signaling in IPF, not only in the epithelial cells but in the fibroblasts. And so we're interested, if these same principles apply in human lung, then could it be that these perturbations in Wnt signaling are disrupting the homeostasis and the ability of the lung to repair effectively? So with that, I'm going to just end, and I'm going to thank most of this Wnt work was by graduate student Ahmad Naban, and Some of the single-cell RNA-seq was performed by Doug Brownfield, a graduate student. I want to thank Roel Noos's lab, particularly Kathy Logan, who was very helpful. And then our in situ technologies was from Monica Negrendon, a postdoc, Dan Riordan, and also my collaborator, Per Harbury, and Andy and Jana are research assistants in the lab. So thank you very much for your attention.